Hello everyone, welcome to VLearn and welcome to our course Molecular Basis of Inheritances. In this course of Molecular Basis of Inheritances, we are going to study about molecules such as DNA and RNA and how these molecules are made. So basically if we talk about the molecular basis of inheritances, you will be having 5 to 8 questions in this molecular basis of inheritances chapter. Let's start the molecular basis of inheritances. If we talk about molecular basis of inheritance, it all started with a scientist named Miescher. So basically, Miescher, the scientist Miescher, was working with pus cell. So he was basically working with these pus cells. From these pus cells, he obtained a substance which was named nuclein. So he obtained a substance from the nucleus of the pus cell and he termed it as nuclein. Further, there was another scientist which was known as Altman. What did Altman do was he studied the chemical nature of nuclein and he discovered that these nuclein are basically acidic in nature. So these are basically acidic in nature. So if we talk about Altman, Altman discovered that nuclein are acidic in nature. So nuclein are acidic in nature. If we talk about this acidic nature, these were therefore called, this substance nuclein were therefore called nucleic acid. These were therefore called nucleic acid. And if we talk about nucleic acid, as you know, nucleic acid act as genetic material. So it act as genetic material in most of the cell. So it is basically the genetic material in all the cell. If we talk about the genetic material, we know that all the cells contain genetic material. And this genetic material can be basically divided into two parts. So if we talk about genetic material can be divided into two parts. So genetic material can be divided into two parts. Genetic material, either it can be DNA or it can be RNA. Genetic material can be of two types, DNA or RNA. If we talk about this genetic material, DNA, in most of the organism, DNA act as genetic material. In most of the cases, DNA act as the genetic material. So in most of the organism, DNA will act as genetic material. But in some of the cases, ribonucleic acid or RNA act as genetic material. So in some of the organism, RNA act as genetic material. If we talk about DNA, so DNA act as genetic material in most of the organism, such as bacterial cell, such as E. coli cells, human cells. Okay. So basically in most of the cases, DNA act as genetic material. But in some of the cases, such as some of the viruses, some virus contain RNA as genetic material. So some virus contain RNA as genetic material. If we talk about, we will discuss in detail the DNA and RNA molecule. If we talk about DNA, so DNA is deoxyribonucleic acid. So DNA is deoxyribonucleic acid. This deoxyribonucleic acid is basically made up of four substances. This deoxyribonucleic acid, or this DNA, is basically the largest biomolecule. This is the largest biomolecule in your cell. And DNA is basically made up of ribonucleotide. Made up of ribonucleotide. So the single unit of DNA is basically deoxyribonucleotide. And you can say that this DNA is basically the polymer of deoxyribonucleotide. So this basically act as polymer of deoxyribonucleotide. If we talk about deoxyribonucleotide. Deoxyribonucleotide is basically consists of three parts, is made up of three parts. The first part of the deoxyribonucleotide is phosphoric acid. If we talk about the structure of the phosphoric acid, the structure of the phosphoric acid contain a phosphate bonded with an hydroxyl group with another hydroxyl group, and it contains two oxygen atom bonded by double bond. If we talk about the second part of the deoxyribonucleotide, a deoxyribonucleotide is basically made up of deoxyribose sugar. Deoxyribose sugar. So it is also made up of deoxyribose sugar. If we talk about ribose sugar is a pentose sugar. Pentose means 5 carbon sugar. So ribose sugar is a 5 carbon sugar. We will study ribose sugar and then we will study deoxyribose sugar. So if we talk about the structure of ribose sugar is a 5 carbon structure and the structure resembles this. So if we talk about, this is the first carbon, this is the first carbon atom, this is the second carbon atom, this is the third carbon atom, this is the fourth carbon atom, and on the fourth carbon atom there is the fifth carbon atom which is attached to an hydroxyl group. So this is basically the fifth carbon atom in the ribose sugar. If we talk about this first carbon is attached to an hydroxyl group, the second carbon and the third carbon. This carbon is attached to the hydroxyl group, this carbon is attached to an hydrogen, this carbon is attached to an hydroxyl group. So this is the basic structure of a ribose sugar. I am not making deoxyribose sugar, I am making ribose sugar. If we look at the structure of deoxyribose sugar, this hydroxyl group is absent. This hydroxyl group is absent. So this is second prime hydroxyl group. And this hydroxyl group is absent in case of deoxyribose sugar. So this is the structure of ribose sugar, and in case of deoxyribose sugar, this hydroxyl group at second carbon is absent. If we further talk about the third structure, the third part of a deoxyribonucleotide is a nitrogenous base. Nitrogenous base. If we talk about the nitrogenous base means nitrogen containing base. Nitrogen containing base. If we talk about nitrogen containing base, these are further divided into two parts, which is purine and pyrimidine. So purine and pyrimidine. Further, we will also see some basic structure of the purine and pyrimidine. So the nitrogenous base, you know that nitrogenous base can be divided into purine or it can be divided into pyrimidines. Purine, if we talk about the purine is a 9-membered ring structure. Purine is a 9-membered ring. Nine-membered ring structure, and this consists of two rings, therefore it is called double ring structure. Therefore it is double ring. The structure is double ring. If we talk about pyrimidine, pyrimidine is a single membered ring structure, and it is basically 6-membered ring. It is 6-membered ring, and it is 9-membered ring. It is double ring structure. It is single ring structure. If we draw the structure of purine, the basic structure of the purine could be looked like this. I am drawing the skeleton structure only. I am not drawing hydrogen bonding part. We will see the hydrogen bonding in next class. So this is the basic structure of a purine molecule. If we count the number, so if we have a look at this structure, the numbering starts from here: first, second, third, fourth, fifth, sixth, seventh, eighth, and ninth. On the position first position, third position, seventh position, and ninth position. On the first 3, 7, and 9 position, there is a nitrogen atom. There is nitrogen present. So on the position 1, 3, 7, and 9 contain nitrogen. Purine can be of two types, that is adenine and guanine. So this is the basic structure. This is the basic structure of purine. This is the basic structure of the purine, and this basic structure is modified into two forms, that is adenine and guanine. If we talk about the pyrimidine, pyrimidine can be further derived into three structure. We will study this structure. These structure are CUT, cytosine, uracil, and thymine. So they can be cytosine, uracil, or thymine. As you know, this is a 6-membered ring structure. This is a 6-membered ring structure. On carbon number 1st and 3, this contain 4 nitrogen. This purine structure contain nitrogen at 4 places, that is 1, 3, 7, and 9. If we number this according in the pyrimidine as well, so this is first, second, third, fourth, fifth, and 6th. So if we talk about the first carbon and third carbon, there will be nitrogenous base. On this structure, that is pyrimidine, there will be first, and on third carbon there will be present nitrogen. So this is all about the basic structure of the nitrogenous base. In the next class, we will study more detail of these structures. Thanks for watching the video, and see you in the next video.